Hello all, I'm gonna run through some of the Unit 4 physiology to review it in advance of your exam. And again, as I've mentioned before with this unit, the lecture or physiology part of Unit 4 is certainly more difficult for most students than the previous 3 units. And so you just got to spend some time. A lot of it is you got to learn these step-by-step processes that occur with, how a motor neuron generates its electrical impulse or action potential and how that leads to contraction of a skeletal muscle fiber. So you got a step-by-step story that you have to learn. Use the study guide that I have posted. It's got lots of fill in the blanks with it that can kind of help walk you through the whole story and what you need to know. If you're having any problems with that, use the discussion board to post any questions you might have about completing that exercise. So I've got some example questions and other things that we'll do here to review some of this material. This is a good preview for Biology 202. When you get to Biology 202, there's a lot more physiology than anatomy in that class, so it's a lot more about the function of the body systems you study in Biology 202 than the parts. And there are a lot of these step-by-step processes and stories that you have to learn to actually understand what's going on with a particular body system. All right, first couple of questions here, though, we're just going to review some basic stuff about muscle tissue and structure. First question here, which type of tissue consists of muscle fibers that are branched and striated? So you want to go back and review just the key characteristics about your 3 different types of muscle tissue. And if you do that, now the keyword striated there, remember that means striped. And so that's the appearance that a couple of types of muscle tissue have when you look at them under the microscope because of the abundance of these protein fibers inside them and how they're all structured together. It gives the 2 of these types of tissues a striated appearance. And hopefully you're thinking skeletal and cardiac for that. Those are the 2 that are striated. But then only one of those 2 is branched. And by branched, that means that one muscle fiber will have a branch that leads off and connects to another muscle fiber. And so hopefully you're thinking B, cardiac muscle is branched and striated. Skeletal muscle is striated, but it's not branched. And smooth muscle is neither of those 2 things. All right, which type of the following is not a function of skeletal muscle? So go back and review your key functions for skeletal muscle tissue. So if you read through those, A, contraction for movement, hopefully, yeah, that's definitely a function of skeletal muscle. Generation of body heat, yeah, that's actually that's not something you think of right offhand, but shivering is one of the key ways that the body warms itself. And remember, when you do cellular respiration, that gives off heat. When you're shivering, skeletal muscles are, without you thinking about it, doing lots of contractions. So cellular respiration is providing the energy for that, and a byproduct of cellular respiration is heat. So these involuntary skeletal muscle contractions that we know of as shivering actually is able to, by breaking down nutrients, releasing energy from those nutrients, heat is given off from those processes. Maintaining posture, yes, your muscles are certainly critical for that. So that leaves us with D, causing joint movements through pushing actions. That's your answer. And now when you first read that, that might sound reasonable, but remember, muscles pull on bones. Muscles, you have tendons that connect muscles to bones, and when a muscle contracts or shortens, it pulls on the bone. Muscles don't push. Sometimes when you look at animations of muscle movements, it might look like a particular muscle is both pulling and then pushing, pulling and then pushing, but that's not the case. Whenever you see an animation of the action of a particular muscle, it's doing the pulling part. It's not doing the pushing part. All right, skeletal muscle structure. So be sure you go back and review the different parts of a typical skeletal muscle, and that includes the connective tissue linings that exist surrounding different parts of the muscle. All right, hopefully my little stylus is going to come on here. Well, this thing is slow for some reason. All right, writing on the iPad, you never know how clear it's going to come out. But remember, this is a diagram from your textbook of we've got a whole muscle that has been cut open. And remember, you have a skeletal muscle composed of skeletal muscle tissue, but you do have connective tissue covering. So remember, like they're showing you here, that kind of white film that you see there, that is the epimysium, the connective tissues that, you can see the stylus is still not going to work too well on this iPad. The epimysium is the connective tissue covering around a whole skeletal muscle, and it blends into and helps form the tendons that you see, the dense connective tissue that attach muscles to bones. And then it's also going to blend in with the periosteum. They're kind of showing you that over here. Remember, bones have a periosteum that surrounds them. And so the epimysium on muscles, that's a connective tissue, a dense connective tissue that more or less just blends in with the periosteum surrounding the bones that the muscles are attached to. All right, then you can divide up a skeletal muscle into subunits. And one of those you see there, that's called a fascicle, that whole thing. And then a fascicle is basically a bundle of muscle fibers. All right, so right here, this little thing that they're pulling out is a single muscle fiber. And remember, a muscle fiber is a cell, even though it has an elongated, strange-looking appearance. And that's the smallest part of a muscle that's actually a cell. When we go below this level, we're talking about the internal parts of a single muscle fiber or a muscle cell. And so we're talking about molecules at that point and not individual cells. So there's a muscle fiber. A fascicle is a bundle of muscle fibers. And then remember, you do have a connective tissue that surrounds a fascicle. That's called a perimysium. And I'm not going to write all these out since the stylus is not writing so well on the iPad. And then remember, each individual muscle fiber, like you see here, it has a delicate connective tissue covering around it, and that's called an endomysium. Oops, I think I skipped a slide. Let me go back. Okay. All right, now here we're zooming in more closely on an individual muscle cell and really just looking at a section of an individual muscle fiber. It's been cut. But notice purple things, those are nuclei. So individual muscle cells will have multiple nuclei. That's not typical of most cells in the body. And the membrane surrounding an individual muscle fiber has a special name, sarcolemma. Sarco a lot of times refers to muscle, and lemma here means membrane. And then outward from there, you're going to have that endomysium you were talking about. So the muscle fiber has its own cell membrane. That's the sarcolemma. Outward from there, you have the endomysium. These things that look like little candy cigarettes or something that are coming out of the muscle fiber here, each of those is a myofibril. Okay, now a myofibril is not the same thing as a muscle fiber. I know some of this terminology gets kind of hairy at times because myofibril sounds like muscle fiber using clinical terminology, but a myofibril is a bundle of a bunch of these proteins that are involved in muscle contraction, your actin and your myosin and your troponin and your tropomyosin, all those nice things that we've learned about. So each of those things is a myofibril. The myofibrils are the locations of actual contraction where that takes place. All right, and then remember, a myofibril is broken up into multiple sarcomeres. A sarcomere is the smallest contractile unit. This is where the business of muscle contraction is taking place. And an individual sarcomere extends. If you've read in your textbook, you've read about the Z lines. And oh, come on, stylus. One of these days, they'll actually invent a stylus that can truly write properly on an iPad. I don't think it's going to happen anytime soon. right, apparently that's just not going to happen. It'll right over there for some reason, but it won't right over here. There we go. All right, between those two zigzagged blue lines, those are called Z lines, and those are anchoring points for the protein fibers within a sarcomere. But everything between those two zigzagged blue lines there is considered to be a sarcomere. So a whole myofibril is just a bunch of repeating sarcomere units. And then if you zoom inside an individual sarcomere, that's what we're looking at down here, you will see that what you predominantly have in there, and keep in mind these are three-dimensional structures. A lot of times they're drawn in 2D like you see down here, but really it's a 3D structure. And they're going to be composed of, in light blue right there, those are the thin filaments that are mostly composed of actin proteins. And then you have, in red there, you have the thick filaments, which are primarily composed of myosin proteins. All right, and then over here on this part of the diagram, what you're looking at is more of a blow-up of a portion of a thin filament there in blue. So all those little things that look like blue squished olives or whatever you want to call them, those are the actin proteins in blue. Remember in brown, the things that look like ribbons, that's tropomyosin, and then in yellow, you have the troponin proteins. And so all of those things are very important for muscle contraction to take place. Then the thick filaments look like this. They're bundles of these individual myosin proteins. There's a myosin protein where it kind of looks like a two-headed snake. And so the myosin heads over here are what actually make contact with the actin thin filaments when contraction is taking place. And then some of the other things you see on there, like the yellow squiggly lines over here and so forth, we didn't really talk about those. Those are proteins that help anchor the thick filaments to the Z line. So there are more details in your textbook about the structure, but we're going to kind of just stick with what we've covered. All right, there's some other important muscle fiber features that you have to know. So again, we're looking inside a skeletal muscle fiber, a single skeletal muscle cell. So all these things that look like red logs that are blown up, those are myofibrils, which are chains of sarcomeres linked together. And now these things you see right there and right there, those are mitochondria. Remember, those are the powerhouses of cells. That's where most of the ATP is generated for cell function, for energy. And so skeletal muscle fibers are going to have lots of those because they bust through a lot of energy to support skeletal muscle fiber contraction. The blue webby stuff you see on there everywhere, that's the sarcoplasmic reticulum, or SR. And the key function for what we've studied with regard to muscle fiber contraction is that's where all the calcium ions are stored or bottled up that are so important for the cross-bridging to take place between actins and myosins. And then in light blue, right here, it's kind of hard to tell on this diagram, but those are the T-tubules. So they are like little indentations of the sarcolemma, the membrane around the muscle fiber that extend inward into the muscle fiber, and they actually wrap themselves around, as you can see, the myofibrils. But those are the T-tubules. I'm just going to write a T. If my stylus will work, come on, there we go. T-tubules there in light blue. So you want to be sure you know these different parts of the skeletal muscle fibers, because they're, if you don't know the parts, then trying to learn the story of muscle fiber contraction is going to be pretty difficult. All right, so now that we're experts on skeletal muscle fiber structure, okay. Here's another question to practice with. Which connective tissue covering blends with the periosteum of bones and or forms rope-like tendons that attach to bones? So basically this is asking us what's the outermost connective tissue covering that we would have around the skeletal muscle. And so hopefully, since we just went over those, you're thinking of epimysium. And you should know what those other words mean. We just went over paramysium and endomysium. What's a ligament? Remember, ligaments are dense connective tissue structures that connect bones to bones. Ligaments connect bones to bones. Tendons connect skeletal muscles to bones. Okay, here's one of those select all that apply type questions. I keep turning my pen off for some stupid reason. It takes about 30 minutes for it to come back on here. Y'all are going to be tired of me fussing about this technology I'm trying to use here by the end of the semester for sure. All right, select all of the following that are parts of the thin filaments. Keyword there, thin filaments in sarcomeres. So we just went over the different parts. So again, when you have a question like this, and this is going to apply not just to your A&P classes, but also in the future when you get into nursing school, if you're going to nursing school, you're going to have lots of these select all that apply type questions. You just want to go down the list and treat each answer almost like it's a true false. So how about acetylcholine? No, acetylcholine is not part of a thin filament. Acetylcholine is a neurotransmitter. That's the signaling molecule that communicates the signal from a motor neuron over to a muscle fiber. How about troponin? Hopefully you're thinking yes on that. Actin, absolutely. Those are the blue beads in the diagram. Myosin, no, myosin is part of a thick filament. Tropomyosin, yes, tropomyosin, that's the ribbon protein we were talking about. Sarcoplasmic reticulum, no, that's the webby-looking membrane-covered compartment that extends throughout the inside of the muscle fiber and kind of wraps itself around the myofibrils. So again, learn the terminology. If this all sounds like a bunch of Greek gobbledygook to you, you got to study those diagrams and learn the terminology in more detail. Sarcoplasmic reticulum, or SR, key thing for what we've studied is that's where the calcium ions that are so critical for muscle contraction to take place, those are stored. They're bottled up in there until they're needed for muscle contraction. Okay. B, C, and E were our correct answers. All right, which of the following are tube-like extensions of the sarcolemma that travel down inside the muscle fiber? And this is not a select all that apply. We've just got one right answer. So again, lots of lingo. If you have not learned the basic terminology, all of this is going to sound like you're taking a test in Spanish. So you got to learn what these words mean. Sarcolemma, that's another name for the cell membrane around a muscle fiber. Then you got these tube-like extensions that travel down. They're like little indentations or invaginations of the cell membrane, the sarcolemma, that go down inside the muscle fiber. And so hopefully you're thinking T-tubule on those. And if you have not done so yet, be sure you watch all those animations from your textbook that are linked on Blackboard. So much of this is way easier to understand when you see it in an animated form than if you're just trying to listen to like a PowerPoint lecture or reading it in the textbook or looking at a two-dimensional diagram. The animations really, really, really help. So be sure you're using those to help you review. All right, motor neuron structure. All right, so you got lots to learn on how muscle fibers contract in this unit. But then on top of that, muscle fiber contractions don't occur unless motor neurons stimulate them to do so. So another pretty extensive part of Unit 4, we've jumped into learning some of the basics about these neurons, which are the main cell types of the nervous system and how they function and their basic parts. So motor neuron structure, you want to go back through all of the required information, the motor neuron parts and so forth for you to know. Remember, the main part of a neuron is called the cell body. All these little smaller appendages that you see everywhere, those are dendrites. And of course, you've got the one long appendage that extends from the cell body down toward, when we're thinking about, now in the next unit you're going to learn about how neurons stimulate other neurons in more detail, but for this unit we're mainly just focusing in on how motor neurons stimulate muscle fibers to contract. So we can imagine here's a muscle fiber over here that's being contacted by this motor neuron. So the long appendage is an axon. And then remember the axon can form branches like you see there. And those branches are all going to extend outward. They all contact the same muscle fiber, but or they all contact muscle fibers within the same skeletal muscle, but you can have some branches there that extend outward to different muscle fibers within the same skeletal muscle. And then on the ends, they have a little knobby area. Those are called synaptic knobs or axon terminals. Can't really see in this diagram too well. So you have that wider area at each of those terminals along those branches. In blue here, those are called Schwann cells. And Schwann cells make a substance called myelin that kind of wraps, basically just kind of like the Schwann cell membrane has just been laid out and wraps itself around the axon. And remember, you got some gaps between those Schwann cells where you don't have myelin, and those gaps are called the nodes of Ranvier. And those are really important for the action potential or the nerve impulse that goes from end to end along the axon. All right, another key place right here where the axon gets wider, where it connects into the cell body, that is called the axon hillock. So that's another thing you need to be aware of as well. Of course, this is the nucleus, but I think that pretty much covers some of the major key things we're going to hear about here in this unit as we're studying motor neurons. And here's another diagram. All right, so again, this unit, if you're just trying to learn terminology, words, and not looking at the pictures and getting a mental picture of what's going on, it's going to be very difficult for you. So you want to study these diagrams and animations and so forth very carefully. So what we're seeing here, this is a slice through the spinal cord that we're seeing there. And the reason that's on there is because that's where the cell bodies of motor neurons live. The cell bodies, that main cell part, those are sitting in the, most of them are sitting in the spinal cord. And you'll learn more about the spinal cord in Unit 5. So on this diagram here, you see that what we're seeing here are two motor neurons. Oh, come on, pen, there we go. And the little dots in the spinal cord, those represent the cell bodies. And then these long lines that you see extending outward, those represent the axons. And then the axons have branches on them, as you see here in the picture. And the different branches may extend to different muscle fibers, but those muscle fibers are all going to be within the same skeletal muscle. So for instance, this skeletal muscle that we're looking at here, this could be the biceps brachii, or the triceps brachii, or the vastus medialis. It's going to be a single muscle. And so what that means is an individual motor neuron is going to stimulate muscle fibers that are all part of the same muscle. You're not going to have a motor neuron that is controlling the biceps brachii and the brachialis. So that's a key thing to keep in mind as well. So what you're looking at here in this diagram is you've got two different motor neurons, and they're both sending signals to individual muscle fibers that you see over here within the same skeletal muscle. Okay, and one of them, if you look closely on the diagram, the one that's represented by the red or the pinkish kind of line there, it's actually going to be sending action potentials to 3 muscle fibers within that muscle. And the purple one is going to be sending impulses to 2 muscle fibers within that skeletal muscle. All right, and you may remember there's a term for a motor neuron and all of the muscle fibers that it controls. Remember that? That's called a motor unit. That came a little bit later in Unit 4, but that's an important term to remember as well. A motor unit is a single motor neuron and all of the muscle fibers that it controls, that it's going to be sending impulses to. So motor neuron, motor unit, for example, here, the red one here, you've got the red neuron, and notice it is contacting that muscle fiber, that one, and this one right here, if you can look close. This diagram's in your textbook. So those things together comprise one motor unit. Okay, and then over here on the other part of the diagram, all right, remember this, this should look pretty familiar. That is an axon terminal or a synaptic knob, so that's at the very end of an axon. And over here in pink, this is all, we're looking at part of a muscle fiber here, and here's the sarcolemma. And where the axon terminal or synaptic knob and that muscle fiber are coming together, you've got a little indentation in the muscle fiber that's called the synaptic cleft. And remember this little gap between the two, the axon terminal and the muscle fiber membrane don't actually physically make contact. That's called a synapse. And some other things here on the diagram. You've got in purple on the muscle fiber membrane or your sarcolemma there, those are the acetylcholine receptors, the little green spheres that you see here in the synapse, and also stored up in here inside these vesicles. That's your acetylcholine neurotransmitter. And when an action potential, that nerve impulse, makes its way down to the synaptic knob, that triggers the acetylcholine to come out. It goes into the synapse, attaches to the acetylcholine receptors, and then that sets off the chain reaction of events in the muscle fiber that leads to contraction. All right, and then membrane polarization. Well, that sounds like a bunch of gobbledygook, but remember, one of the key things when we're learning about muscle fiber and motor neuron function in this unit is that we have to generate these electrical impulses that lead to this signaling and these changes that take place. And in order for all that to work, the cell membranes of our motor neurons and our muscle fibers have to be polarized. And again, what that means is that there is a charge difference on one side of the cell membrane versus another. So if we're zooming in here on part of a motor neuron cell membrane, we have learned that you have various ions, positively charged and negatively charged substances on one side of the membrane versus the other. But under normal circumstances, if you totaled up all those charges, the inside of a cell membrane, motor neuron or skeletal muscle fiber, tends to be more negative than the outside. Okay, and we can actually measure that as a voltage. So it's measured in millivolts. Minus 70 millivolts is sometimes called the resting membrane potential. I know some of this terminology can be pretty crazy, but all that means is that is a numerical measurement of how much more negative is the inside of the cell membrane versus the outside. And so we would say that that cell membrane is polarized. In a typical motor neuron in the human body, it's about minus 70 millivolts. And then we also learned a little bit about how that polarization is maintained. And you guys can go back and take a look at some of these things. Animations, again, are generally the most helpful, but along these cell membranes you have potassium leak channels, and you got sodium leak channels, and then you have proteins that are really important called the sodium potassium pumps. Stylus is not going to work. Okay. Another key thing that we learned about, this is something you definitely want to keep in mind. On the inside of a cell membrane at rest, you tend to have more potassium inside than outside, and the reverse is true for sodium. You tend to have a lot more sodium ions, or the concentration of sodium ions is higher outside the cell membrane than inside. And what these leak channels are doing, this means these are little channels, they're proteins in the cell membrane that allow ions to leak out. And these ions are going to move by diffusion, so they're going to move from areas where they're more highly concentrated to less. So these potassium leak channels are constantly allowing some potassium. Potassium's more concentrated inside the cell, so it's going to tend to, if you have a channel that's letting it leak out, it's going to move out of the cell. So the cell is losing positive charges, which that's one of the key things that keeps the inside a little bit more negatively charged. If you're losing positive charge, you're going to become more negatively charged overall. Sodium leak channels, though, do the opposite. Since sodium is more concentrated outside than inside, they're going to allow sodium ions to kind of gradually leak from outside to inside. And you might be thinking, well, sodium is positively charged too, so that's going to reverse what the potassium leak channels are accomplishing. And that's true. So the sodium leak channels are helping to keep things balanced so that the inside doesn't become too negative. We want to keep it right around minus 70 millivolts. All this stuff has to be very carefully coordinated. And then over here, these sodium potassium pumps are really, really important. Those are, a pump protein is a protein that uses energy to force things to move across the cell membrane in the opposite direction of how they would naturally want to go. So the sodium potassium pumps pump sodium out and pump potassium in. So they pump against the concentration gradient. Sodium naturally wants to flow from outside to inside through a channel. So if you're going to move it out, you got to pump it out. You got to force it out. And the same thing for potassium. You got to force it inside against its concentration. And the sodium potassium pump requires energy. It gets that energy from ATP, and they actually use up quite a bit of the ATP that's generated by these motor neurons. So all those things are working together to keep the cell membrane of a resting motor neuron polarized. An action potential is a temporary but very rapid reversal in membrane polarization. All right, and on a graph, it would look like this. So if you're going to generate an action potential, remember the resting membrane is sitting here at minus 70 millivolts. Then we have another important voltage, minus 55, and up here about plus 30. And so resting membrane potential is minus 70. With an action potential, what happens? We studied in this unit that when a motor neuron is getting stimulated, like it's being told, hey, you need to generate an action potential because we need the skeletal muscle that you're controlling to contract. So that kind of information is coming down from the brain. And so you'll have little local areas of a motor neuron where that's happening. And when we say that the motor neuron is getting stimulated, that means the voltage is going up along little local areas. So you have some sodium that's flowing in to make the inside of the membrane become less negative. And if you go back and you visualize a motor neuron and what it looks like, so that can be happening on the dendrites and on the cell body. You're getting these positive charges coming inside the motor neuron. The key place that's critical for an action potential is the axon hillock. So at the axon hillock, if you get enough of this stimulation, and again, by stimulation, that generally means sodium ions are coming in through these channels. If at the axon hillock, you reach minus 55 millivolts. Okay, then you've got some what we call voltage-gated sodium channels that swing open. And they only open at minus 55, so you've already got to have a partial depolarization going on, a partial change in membrane voltage that's already happened there. But if it hits minus 55, you got enough sodium that came in to raise it up to minus 55. You've got these voltage-gated channels that swing open, and they allow lots of sodium ions to rapidly come in. The cell membrane rapidly reverses its polarization. It becomes depolarized. You go up to plus 30 because all these sodiums rushed into the cell. Then those channels close, and then you've got some potassium voltage-gated channels that open. And what they do is they start letting, so you had all this sodium that came into the cell, and that's what allowed us to go up to plus 30. These voltage-gated potassium channels swing open, and they allow, remember, potassium is going to flow through a channel from inside the cell to outside. So all this potassium goes out, and that's called repolarization. You're repolarizing the membrane. Then your good old sodium-potassium pump kicks in and restores everything back to normal in that area. And notice temporarily the membrane there becomes hyperpolarized, which means it's below minus 70. So this is an action potential is a temporary, very rapid flip-flop in the membrane voltage. And it starts at the axon hillock, and then as we studied in this unit, once it starts at the axon hillock, it's going to very rapidly travel all the way down the axon from one end axon hillock all the way down to those axon terminals or synaptic knobs down at the other end. And so you want to go back and review. I know some of this can be a little bit difficult to follow when you're first studying it, but you want to go back and review how all that works. Watch the animations. They're extremely helpful. All right, and remember the axons of motor neurons are myelinated, the ones that control skeletal muscles. Now, when we get to the nervous system, Unit 5, you're going to learn that not all axons have myelin around them. But the myelin shown there in blue, it's made by the Schwann cells that just live along the axons of motor neurons. It's an insulation. And the action potential, okay, we said it starts at the axon hillock, and then it gets regenerated at each node of Ranvier. So it basically skips the parts of the axon that are covered in the myelin. You don't have to regenerate the action potential at those places. Go back and look, an action potential moves from one part of the membrane to the next, to the next, to the next, and it moves in one direction. It doesn't move both ways. That's really critical for its function. But when you have a part of the axon that's wrapped up in myelin, the action potential skips basically from there to the next place, to the next place, and so forth. And that helps it travel much more rapidly down the axon. All right, so a few questions about all this membrane potential and action potential gobbledygook that we've been talking about. Let's try this one. Along a non-excited resting portion of a motor neuron membrane, which ions are more concentrated inside the cell? All right, and hopefully you're thinking potassium, or potassium abbreviated with a K and a positive charge. Which one is more concentrated outside the cell? Hopefully you're thinking sodium. All right, what happens when threshold is reached along a portion of a motor neuron membrane? All right, so this is one of those where you got to know the terminology, or this is going to sound like a foreign language to you. Threshold, remember we were looking at the action potential graph. resting motor neuron membrane, you're starting off at minus 70. Threshold is about minus 55. And so if you have depolarization, so the charge along the cell membrane is becoming a little bit more positive, once it hits minus 55, and this is going to start at the axon hillock, that's called threshold. And so what's going to happen? So if you read through those, you can pause the video if you want to take a couple of minutes to read through those. The correct answer is going to be C. And again, the reason it's C, voltage-gated sodium channels open. A little picture disappeared on me. So we started out down here at minus 70. When you hit minus 55, that's called threshold. Voltage-gated sodium channels open. That lets lots of sodium inside that little local area of the membrane, and your voltage will go all the way up to about plus 30. That's called depolarization of the cell membrane. Okay, next question. Why does the membrane of a motor neuron become temporarily hyperpolarized at the end of an action potential? So again, what in the world does that mean? So if we graph our action potential, all right, you start out at minus 70, you hit minus 55, that's threshold. Boom, sodium rushes in because you got those voltage-gated sodium channels that open. You go up to about plus 30. They close. And then voltage-gated potassium channels open. You got more potassium inside than outside. So they start letting all this positively charged potassium out. All right, and your membrane, the voltage across the membrane, temporarily drops below minus 70. All right, that's called hyperpolarized. And the reason for that, if you're looking on there, hopefully you have picked answer A. Excess potassium ions flow out and make the inside more negatively charged than normal. Okay, and that's what brings us temporarily below minus 70. That's called hyperpolarization. All right, and when a cell membrane is hyperpolarized, it makes it more difficult for it to reach threshold. Okay, so this is one of the things that helps make sure the action potential just moves in one direction. It's not also backing up and going this way. We want it to go from the axon hillock towards the axon terminals down at the other end. All right, and then when an action potential makes it all the way down to the axon terminals, so again, that's what we're seeing right in here. We're just looking at the very end of a axon, and it's divided up into little branches, and here we're zooming in just on an axon terminal where it's forming a synapse with a muscle fiber membrane. And remember, as continue our story, action potential makes it all the way down that nerve impulse to the synaptic knob. Remember, okay, that opens up calcium channels. So on this diagram, these little purple things right there and right there, those are calcium channels. Calcium ions have a double positive charge. And those calcium channels let calcium come inside the synaptic knob. Calcium in the synaptic knob causes acetylcholine, which is abbreviated as ACH. That's stored inside these little vesicles, these little membrane-covered compartments. In here, the little green dots. Calcium triggers those vesicles to travel up to the surface of the axon terminal or synaptic knob that releases the acetylcholine into the synapse. The acetylcholines, the little green dots, travel across and they attach to, over here in purple, you've got acetylcholine receptors. When that attachment occurs, that opens a channel, and kind of the key thing here is that that lets sodium come inside the muscle fiber, sodium ions. All right, and so that should remind you of what we were just talking about with the motor neuron. When the sodium ions are coming in, what's going to happen along the muscle fiber membrane if we were graphing it out? If you're starting off at -70 down here, you're letting sodium in, so the inside of the muscle fiber membrane becomes less negatively charged. And if you hit threshold, boom, you're going to have an action potential on the muscle fiber membrane. It's going to go in both directions away from the axon terminal. You're going to have an action potential just like you did along the motor neuron. All right, and then why in the world do we need to do that? This is very critical in order for the action. Remember, we're trying to get to muscle fiber contraction. The whole point of all of this rigmarole is that we're going to actually make a skeletal muscle fiber contract. And so what they're showing you here in this diagram from your textbook, that's that little pink line there is supposed to represent the action potential, this electrical impulse that travels across the muscle fiber membrane, or your sarcolemma. That's another name for that. And like they're showing you here, it goes down inside those T-tubules that we learned about, those little extensions of the cell membrane that go down inside the muscle fiber. Those T-tubules are located up against the SR compartments. And this is all blown up for us over here. So in pink there, that's showing an action potential coming down into a T-tubule. And as that happens, that winds up triggering, as that action potential travels along the T-tubule, that actually triggers calcium ions come out of the SR. Calcium comes out. So all these little things you see over here, those are calcium ions. All right, and then why is that important? That's going to lead to, when calcium ions come out of the SR, that is what will lead to the actins and the myosins forming their cross bridges. And so like they're showing you here on the diagram, when a muscle fiber is at rest, the tropomyosins, that brown ribbon protein, they're draped over the binding sites for the myosin heads. When calcium is released from the SR, the calcium attaches to troponin, the yellow protein that you see there in yellow. That causes the troponin to drag the tropomyosin ribbons away from those binding sites on the actins. So it looks like you can now see the little olive pits get exposed. And again, you got to have calcium out of the SR for that to happen. Why did the calcium come out of the SR? Because you had an action potential generated along the muscle fiber membrane. Why did that happen? Because the motor neuron generated an action potential that caused acetylcholine to be released into the synapse, which triggered the action potential along the muscle fiber membrane. So remember, you're looking at a step-by-step story here, and all of these things are critical. All right, now once that has happened, the myosin heads there can actually make physical contact with the actins, and they're going to do their power stroke like that. And that's what actually leads to muscle fiber contraction. All right, and those events are called the cross-bridge cycle. And what that's telling us is this is, as long as calcium is out of the SR, cross-bridging will continue to take place. And so like they're showing you what they're starting with, you got to start the cycle at some point. Myosin heads are, hang on just a second. All right, sorry about that, I had a temporary interruption. Okay, so back to our cross-bridge cycle here. Notice you got the myosin head, and it's attached to the actin in blue. And the reason those two can come together is because the tropomyosin ribbon has been pulled away by troponin in yellow. And the reason that was possible was because the calcium ions that came out of the SR attached to the troponin. So you got to think through these step-by-step processes. All right, and the other thing that's important here is ATP. So these two little things there that look like eggs, those represent ADP and phosphate. You know, they've already been split apart since we're starting at this point of the cross-bridge cycle. Notice they come off right there. And the myosin head does its power stroke. It goes like this. And that's what's causing the actual contraction to take place as this cross-bridge cycle occurs over and over. And then in the next part, this little thing that looks like a starburst here, that's actually ATP. So an ATP molecule attaches to the myosin head. That allows the myosin head to come off like they're showing you there. And then ATP gets hydrolyzed, which means it gets broken apart into an ADP molecule and a phosphate, sometimes abbreviated as PI, for inorganic phosphate because it's an inorganic molecule. And as that happens, that releases energy. When ATP gets broken apart, it releases energy. That allows the myosin head to get cocked, like you're cocking a trigger on a gun. It takes energy to make that movement happen. Then the myosin head attaches to another actin, and the cycle continues. It's going to do its power stroke. It's going to come off, ATP is going to bind, it's going to come off, ATP gets broken apart into ADP and phosphate, it gets cocked, rebinds to another actin, boom, comes off, gets cocked, reattaches to another actin, and boom. So that's called the cross-bridge cycle. And again, in order for that to keep occurring, you've got to have calcium out of the SR. The only thing that's going to keep your calcium out of the SR is to have action potentials traveling across the muscle fiber membrane. All right, so some questions here about the neuromuscular junction. As the action potential passes across the synaptic knobs of a motor neuron at the axon terminals, blank enter the synaptic knob and triggers the release of blank. So again, now here we're thinking about the synaptic knob. So an action potential has come down the axon, it passes across the synaptic knob membrane like a wave. What did we say will enter the synaptic knob at that point? Hopefully you're thinking calcium ions. You got calcium outside the cell, they come in through channels that open when that action potential arrives. And that triggers, what do we have stored inside the axon terminals that will get released when that happens? Hopefully you're thinking acetylcholine. That gets released into the synapse when that occurs. We're down here in our synapse. All right, next question. Blank are stored in the sarcoplasmic reticulum. Okay, so let's just think about that one. What did we just talk about? What's stored in the sarcoplasmic reticulum? That's in the muscle fiber. Calcium ions. Hopefully you're thinking calcium ions. And those calcium ions then bind to what on thin filaments when they are released? Hopefully you're thinking troponin. Remember, calcium attaches to troponin, then troponin changes shape and it pulls the tropomyosin ribbon away from those binding sites that you have on your actins. All right, how about this? What binds the myosin heads and causes them to detach from actin in order to start a new cycle of cross-bridging? So you've got a myosin that's attached to actin. What has to attach to the myosin head to make it come off? And hopefully you're thinking ATP. You got to have an ATP because that's going to provide the energy when it gets broken apart to cock the myosin head, put it back in the cocked position so that we can keep our contraction going. All right, another question here. Bubba is doing dumbbell arm curls to try to jack up his biceps and impress a hot chick. As he raises the weight and flexes his elbow, a blank contraction is occurring. As he lowers the weight, a blank is occurring. All right, so we learned about some different kinds of muscle contractions. So if you're just kind of thinking about it in basic terms, when a muscle contracts, we're thinking about the muscle shortening. And that's what's happening. Like if you're doing dumbbell curls, you're flexing your elbow like this. The biceps brachii is the prime mover, the main muscle that causes you to flex your elbow if you go back and review your muscle actions. All right, so in that case, yeah, the muscle itself is contracting, and that's occurring down inside those muscle fibers and your myofibrils, your sarcomeres. But the actual, if you're thinking about the whole muscle itself, it's actually physically shortening at that point. So there was a name for that type of contraction, and hopefully you're thinking concentric for contracting or shortening of the muscle. But then if you think about it, if you were holding a heavy dumbbell and you're lowering the weight, your biceps brachii muscle is getting longer, it's lengthening itself if you're just looking at it whole muscle type view. But down inside that muscle, contraction is still taking place. That muscle is still, actins and myosins are still cross-bridging, because if they weren't, you would drop that weight on the floor. So you can still contract a muscle even though the whole muscle itself is actually stretching out and elongating, like when you're lowering a heavy weight and extending the elbow. So that type of contraction has a name as well, and hopefully you are thinking about eccentric contraction. The eccentric is when the muscle is lengthening even though you're still contracting it. And there was one more term there that we learned with regard to muscle contractions, and that was isometric. Remember, an isometric contraction, if I was holding a heavy weight in my hand and not moving it, not doing a concentric contraction, not doing an eccentric contraction, but just holding it stable, that is called an isometric contraction. And you may have heard before of isometric exercises. All right, then we also learned about some things that will increase muscle contraction force. So this is one of those select all that applies we want to go through. How about A, more frequent action potentials generated by the motor neurons controlling the muscle? Hopefully you're thinking yes on that. If you go back, in order for you to do a muscle contraction, like if you were doing bicep curls or running or whatever, it's not like you get one action potential on a motor neuron leading to one round of cross-bridging taking place. In order for you to do sustained contractions using a particular muscle for a longer period of time, motor neuron has to send more and more and more and more action potentials over and over. And why is that? Because that's going to stimulate acetylcholine to keep coming out of the motor neuron. That's going to continue to trigger action potentials on your muscle fiber. That's going to keep calcium out of the SR, and that's going to keep it available so that you can keep doing your cross-bridging. Now remember, we also took a look at what happens with muscle fiber contractions. So you can have what's known as a twitch, like one action potential from a motor neuron leads to one round of shortening and then relaxation. That's called a twitch. But then if you have another action potential from a motor neuron followed very quickly by another, by another, by another, by another, you get that stair stepping, and eventually you reach what's called tetany. And that's where the skeletal muscle is in a permanent state of maximum contraction until it winds up becoming exhausted. That's called tetany. And in order for that to happen, you have to have repeated motor neuron action potentials being generated over and over and over again. All right, so more frequent action potentials by a motor neuron will lead to increased muscle contraction force. How about a reduction in oxygen gas availability? Hopefully you're thinking no on that. Why do we need oxygen gas? We need it for cellular respiration. Cellular respiration is critical for making ATP. You got to have ATP to do muscle fiber contraction. So that's going to be a no. Less oxygen gas. That's just kind of common sense in a way. You're not going to be able to contract your muscles as forcefully. Optimal muscle length prior to contraction starting. That's a yes. If you go back and review, we talked a little bit about how, and here you're not talking about a contracted muscle, but just the muscle itself before it starts contracting. If there's an optimal length, and by optimal length, that means like you're down deep inside that muscle in the individual muscle fibers, your actins and your myosins are overlapped just enough so that they have plenty of room to slide across each other and give you a good contraction force. If a muscle is too stretched out before the muscle fibers start contracting, your actins and your myosins might not be overlapping enough. They can't even make physical contact with each other if that muscle is too stretched out when you start a round of contraction. Or if your muscle is shortened and your actins and your myosins, they're not making contact yet, but when they're starting off, if they're already overlapping each other like this, when they do start making contact, they don't have as much room to slide across each other. So you want to have an optimal, in order for maximum contraction force, you want to have an optimum muscle length before contraction actually begins. And a couple of examples of that, if you tried to lift something heavy and you were starting with your elbow already flexed, that biceps brachii muscle is already shortened, you can't pick up as heavy of a weight starting off like this as if your elbow was more extended. Or like when you are trying to use your hamstring muscles to climb up 2 stairs at a time instead of just 1 stair at a time, and doing that leg extension that's required. You can't do that as well going up 2 stairs at a time as 1 stair at a time, and that's because the muscles are not at optimal length. They're too stretched out before you start doing that. Recruitment of smaller motor units. That's actually a no. Remember, a motor unit is a single motor neuron plus all the motor fiber, all of the muscle fibers it controls. Smaller motor units have smaller numbers of muscle fibers in them. Larger motor units have larger numbers of muscle fibers in them. So it makes sense that you would want to recruit or use larger motor units for more forceful contractions. You're going to have more and generally larger muscle fibers involved. Recruitment means like the brain is recruiting or telling motor neuron, stimulate your muscle fibers to contract. So if it's a motor neuron that's in control of more muscle fibers, that's going to generate a stronger contraction. All right, E, contraction of more muscle fibers within a muscle. Yes, if you want a stronger contraction, you want to contract more of the individual muscle fibers within that muscle. Remember, a whole muscle is made up of tons and tons and tons of muscle fibers. They don't all contract at the same time. And the brain can recruit more or less of those to contract at any given moment. Contraction of muscle fibers with larger diameters. Yes, muscle fibers with larger diameters are going to have more actin and myosin inside them, so there's going to be more cross-bridging going on. They're going to be able to generate more forceful contractions. So there we had A, C, E, and F as an answer. Muscle tone, here's a multiple choice. Muscle tone refers to which of these? Now you might jump at C, muscles with a healthy appearance due to regular exercise and low body fat levels, but that's not what muscle tone means in muscle physiology. It means B, the constant, slightly contracted state of all muscles. So when we say a muscle is relaxed, really their muscles have some low-level contraction going on all at the same time. If that was not happening, we would have no posture. We would just be like a limp rag doll all the time. So your muscles, that's called tone, this kind of low level of contraction that takes place even when we are considering a muscle to be resting. There's going to be some low-level contraction occurring. Energy for muscle contractions. We talked about some of the ways that your skeletal muscles generate all this ATP that they need in order to sustain contraction. And go back and look at why. So we've already talked about how ATP is needed for the cross bridging to take place. That's going to use up lots of ATP. What are some other reasons why we need ATP? We talked about how you need ATP for your for action potentials to be generated to maintain the proper voltage across membranes, and then we also talked about how ATP is needed when you no longer need that calcium around to cause cross bridging to take place. It has to get bottled back up into the SR, and so ATP is required for that as well. That's energy that's needed to pump that calcium back into the SR. That's another reason why you need ATP as well. So in a nutshell, ATP is absolutely critical for muscle fiber contraction. All right, what process allows for maximal ATP production and endurance? So key word there is endurance. If you're thinking about an endurance runner, what method is that endurance runner using to generate most of the ATP needed for muscle contraction? And hopefully you're thinking here aerobic cellular respiration. That's the process that generates the most ATP. So starting with a certain amount of glucose or other nutrients, you're going to get the most bang for your buck. You're going to make the most ATP, harness the most energy by doing aerobic cellular respiration. The problem is it's a longer process. It's a more complicated process, so it's not the most rapid one. So when you need energy very quickly, you need ATP very quickly for like lifting a heavy weight for a short period of time or running a sprint, you've got some other pathways available. The anaerobic pathway, anaerobic means oxygen gas is not required, but that's a temporary process, where you can make ATP starting from creatine phosphate. Go back and review what we talked about with that. That's another process for temporarily providing energy to make ATP. And then also your skeletal muscle fibers store glycogen. Glycogen is a polymer or a chain of glucoses that can get broken apart. Then those glucoses feed into, over here, cellular respiration or this anaerobic pathway to help provide ATP that's needed for muscle fiber contraction. All right, this substance is made as a waste product in muscles that use the anaerobic pathway to make ATP and causes a burning sensation. So it's like a waste product. You're breaking down glucose to make ATP just like you do with cellular respiration, but the process stops after glycolysis, so you don't make as much ATP, and then you generate a waste product from that, and hopefully you're thinking lactic acid on that. Now, lactic acid, you can't tolerate a huge buildup of that. It irritates sensory nerves that are involved in pain sensation, so you start to feel that burn from using a muscle, and you can't tolerate that for very long. All right, which of the following are true about an elite marathon runner? Select all that apply. All right, so let's go through these. So again, this is going to be an endurance runner. Likely has more fast glycolytic or white fibers than other types of muscle fibers. Fast glycolytic. And hopefully you're thinking no on that. We talked about how those types of muscle fibers are involved more in sprints or temporary bursts of heavy, forceful contraction. Like if you're lifting a heavy weight or running a sprint, your fast glycolytic or white fibers get involved in that. And the reason they're white is because they lack myoglobin and they don't have as much of a good blood supply, which means they don't have as much oxygen gas available, so they can't do aerobic cellular respiration as much. B, muscles excel at using the aerobic pathway to generate ATP. Yes, endurance, think aerobic pathway for making ATP. Training has caused hypertrophy of quadriceps and hamstring muscles. What is hypertrophy? That means an overgrowth of the muscles. The muscles have gotten larger. The muscle fibers within the muscles have expanded in size. You've They've made more actin and myosin filaments for cross-bridging, for more forceful contractions. Yes. Hopefully you're thinking yes on that. Generally, endurance runners will have hypertrophied or larger leg muscles. Her red muscle fibers contain abundant myoglobin and mitochondria. Hopefully you're thinking yes on that. Red muscle fibers, these oxidative muscle fibers that are really good at doing aerobic cellular respiration, they contain lots of myoglobin. Myoglobin is a protein that helps store oxygen inside the muscle fibers, and it has a reddish appearance, kind of like your red blood cells, which you'll learn about in Biology 202. And mitochondria are the locations of aerobic cellular respiration. So if a muscle fiber is going to do lots of that, you're going to need lots of mitochondria to support it. E, her leg muscles will likely convert to connective tissues more quickly than those of someone who exercises less often. And hopefully you're thinking no on that. Muscles are kind of use them or you lose them. So as you age, yeah, some of your muscle fibers die if they're not being used frequently enough. There's nothing stimulating them to keep them alive, and then they wind up converting over to connective tissue, and that's why as you get older, your muscles tend to not hypertrophy, but atrophy. That's the opposite, where they tend to kind of wither away, and the skeletal muscle tissue gets replaced with connective tissue. All right, that was the last question. I know you're really sorry about that because this has been a long review, but it's kind of a complicated unit over on the physiology side. And again, those are just some example questions. They're not real test questions. You will be asked about similar types of concepts, though, so you want to keep that in mind as you're studying. And if you come up with anything over the as you're preparing for the exam, don't hesitate to contact me. I prefer, though, that you post things on the discussion board so that other people can benefit from that information as well. So good luck with the test, and then moving on forward from there into Unit 5.